Okay, so I asked you guys to draw the picture of one cell and identify the nucleus. Yep. Labeled a few times. Yeah, the area in the middle, right? The nucleus. Okay. What's that say? Get wrecked. Someone needs to go to English class. That's spelled wrong. Oh, there's a good one. Yeah, it's in the middle. All right, so here's another question. Oops. Does the nucleus always have to be in the middle of the cell? Not always, right? Okay. So we're going to be looking at a lot of cell parts today. Hopefully we'll start our cell project today as well. But we'll be looking at cytoskeleton, sort of what gives the structure of the cell, because the cell doesn't have a skeleton. We'll look at how DNA directs activity in the cytoplasm, where you find it at. We'll look at the organelles involved in protein production, which you already know that, I think. We'll talk about vesicles and vacuoles and how a vacuole is really just a type of a vesicle, and how cells get their energy with a very special organelle called a mitochondrion. So eukaryotic cells have an intricate network of protein fibers called cytoskeleton. And this provides the interior framework of the cell itself. So cells, they don't have an external skeleton, but what they do have is this internal scaffolding of fibers. You guys know what fiberglass is? Yeah, it's got all of these fibers that go in all different directions, and they give that material a very strong structure based on the fact that they have these interwoven fibers. So inside of the cell, we have these proteins that are called cytoskeleton. There's actually two types. One is microtubules. The other one is called microfilaments. And the cytoskeleton helps the cell move. It keeps its shape, and it organizes its parts. It acts as sort of the walls within the cell that separate, let's say, the living room from the dining room. And there are three different kinds of cytoskeleton fibers: microfilaments, microtubules, and then there's the ones that go in between. They're not quite a filament, not quite a tubule, so they're called intermediate fibers. And so if we look at the picture of the cell here, I'm not going to really worry about. Oh, now I need my pen. Shoot. So if I were to label cell parts, what would A be? Just yell it out. What's the structure in the middle of the cell? The nucleus. And then we have these, what are these little holes? Nuclear pores. So we copy DNA onto RNA, which wiggles through to go out into to find, and so B is a ribosome, right? Okay? And ribosomes make proteins by assembling amino acids and forming peptide bonds, right? Okay? And so then we see these long structures. See these microtubules and filaments? So C would be cytoskeleton here. And then if I take a look even closer, I have these other things that are called organelles. And so here I have the endoplasmic reticulum. This is the rough endoplasmic reticulum. And over here I have another organelle called a mitochondria. Okay, mitochondria makes our energy. We'll talk more about that later. You like those? No? And so DNA contains instructions for making proteins which control most of the activity of the cell. Where is our DNA found at? Where do you find your DNA at? Where is DNA stored? You all should be on your own device doing this. Turn it on. We'll figure it out. Probably need some power. Make sure you're using the Chromebook network. I'll help you. That's what I'm telling you. Get her turned on. So I asked, where do you find your nucleus, or where do you find your DNA? Sorry. Yes, in the nucleus. Can DNA ever get out of the nucleus is my next question. Can DNA ever get out of the nucleus? Can DNA ever get out of the nucleus? A big mouse. It's really weird. Did you turn this big mouse thing on?
No.
Oh. Try restarting that. Which number is that? I'm going to have a nice little talk with Ryan Arcangeli because he's the only kid that would do that crap. And he sits right there. Okay, let's see what you came up with. So here's the kicker. It cannot. So DNA is inside of that nucleus. It doesn't get to leave. You wouldn't want it to leave either. If it left, it could get damaged. You don't want to screw up your blueprint of life, right? So DNA is trapped and kept safe inside of that nucleus. So DNA has the instructions that are, we know we copy DNA onto RNA messages, which leave the nucleus through a nuclear pore. They go out in the cytoplasm. They find a ribosome. And that's where we make our proteins at. So we store DNA in our nucleus. Prokaryotes do not have a nucleus, though, right? And so in a prokaryotic situation, their DNA is directly exposed to their environment. There's no nucleus there to keep it safe. So the nucleus is a double membrane structure, and it has the structure that surrounds it called a nuclear envelope or a nuclear membrane. I don't care what you call it, but if I say envelope, no, I'm talking about the nuclear membrane. And nuclear pores are located on that envelope and act as channel to allow certain molecules to move in and out of the nucleus. What is one molecule, one type of molecule that can get through a nuclear pore? Try to connect now. It's working now? Okay. Name one molecule that can get through a nuclear pore. I probably have brainwashed you into remembering this, but so you should probably just go, oh, I know. Could have put a few things. But RNA for sure, right? Some proteins, yep. Cody Bell. You don't think you'd fit? You could have said H2O, right? That would have worked too. Water can get through there, salts, things like that. And then, within the nucleus, there's this dark region called the nucleolus. And this is a structure within the nucleus where ribosome parts are made. And you're probably thinking, well, where do ribosomes come from? Well, something has to make them, right? Well, there's this area inside of the nucleus which makes the things that make our proteins, and it's called the nucleolus. It sounds like nucleus. It's not. It's the nucleolus inside of the nucleus. Usually it's this dark area. And these ribosome parts are transported out of the nucleus into the cytoplasm where they are assembled to form a complete ribosome. So that means there's two parts of every ribosome. There's a large subunit and there's a small subunit, and they come together outside in the cytoplasm so that you can make proteins. But those two subunits are made inside of the nucleus. And then they're shipped out through a pore. Once they get out, they assemble, and then they're waiting for their RNA message to make a protein. So this dark area in the middle is our nucleolus. This is where ribosome parts are made. Of course, we have nuclear pores inside of our nucleus. This yellow material here is your DNA. And this D is the the nuclear membrane or the nuclear envelope. So this yellow stuff that's everywhere is your DNA. We copy DNA onto RNA, which wiggles through to go out into to make a Where do we make ribosomes at? Inside of the nucleolus, inside of the nucleus. Ribosome parts wiggle through a nuclear pore where they assemble to form a ribosome who is waiting for, everybody say RNA, so they can make a protein by assembling amino acids and forming peptide bonds. So each ribosome in a cell is made of RNA and many different proteins. Ribosomes that are suspended in the cytosol are called free ribosomes because they are not attached to anything. You got friends that don't have boyfriends or girlfriends. They're free, right? They can go out, they can do. But as soon as your friend gets a boyfriend or girlfriend, what happens? They can't hang out anymore, right? And then they stop hanging. Anybody have that happen with their friends? Yeah. See, she called you out, right there. All right. So free ribosomes make proteins that remain inside of the cell. So here's the thing: if you're a free-floating ribosome and you're just floating around in the cytoplasm, you're going to make a protein, but that protein will stay in the cell. It's not going to leave. So your cell needs proteins. Those ribosomes that make those proteins are just floating around. They're not attached. Boys. So ribosomes that are attached are what are called bound ribosomes. And they're attached to this structure called the endoplasmic reticulum. And bound ribosomes make proteins that are going to be exported from the cell. And then the ribosomes can switch between being both bound or free depending on what proteins the cell needs to make. So here's the thing. Your cell needs proteins inside of it. Outside of the cell, sometimes your cell has to make proteins that are going to leave. Here's an example of that. I have to make more cytoskeleton inside of my cell. Cytoskeleton is a protein, right, like a microtubule or a filament. So those would be free ribosomes that make those. If I'm going to, let's say, send insulin out into my bloodstream, that means it's going to have to leave the cell. I'd have to make that in what's called a bound ribosome because it needs to be packaged to leave the cell. You get that? It'll make more sense in a minute, I promise. So some proteins that a cell manufactures are needed outside of the cell that makes them. Proteins that are sent outside of the cell are packaged in vesicles. Vesicles are small membrane-bound sacs that enclose the proteins and keep them separate from the rest of the cytoplasm. And the endoplasmic reticulum and the Golgi apparatus are organelles involved in preparing proteins for extracellular export. So I need everybody to do this with me. So if I am a ribosome attached to the endoplasmic reticulum, what type of ribosome am I? Everybody say bound ribosome. So if I'm a ribosome, that means I make a protein by assembling amino acids. And if I'm a bound ribosome, that protein is going to leave the cell. The endoplasmic reticulum that I'm bound to will package it inside of a vesicle. The Golgi apparatus will repackage that again. Well, let's make sense out of that. When you go to the store and buy chips, what do you buy? Do you buy a handful of chips? You buy a bag of chips. That's like a vesicle. You put your chips in a bag, right? Because the factory that made those knew they were going to ship them out, so they put them inside of a vesicle bag. Well, you can't just throw the bags in the truck. What do you got to put the bags in? A box, right? That's like the Golgi apparatus repackaging it again, and then it can send it to the store, right? You get that? That's kind of the way this works a little bit. So the endoplasmic reticulum, or ER, is an extensive system of internal membranes that moves proteins and other substances throughout the cell. You ever go to the bank and you see the sucky tube? It goes, it takes your stuff through and it goes to the other side. You know what I'm talking about? Okay, so that's like the endoplasmic reticulum, except inside of a sucky tube is a little hard shell that sucks it up. It's the, I don't know what it's called, the vacuum tube. And so instead of that hard canister, it's like this little fat bubble that you put your proteins in, and then they get sucked up through the tube, and they get sent to another packaging company. All right. So the membranes of the ER are connected to the outer membrane of the nuclear envelope. That means that this isn't like a separate structure. It's actually connected. It's like an extension of the nucleus. Okay? So inside the nucleus, we copy DNA onto RNA. RNA wiggles through a nuclear pore, and if it finds a ribosome attached to the endoplasmic reticulum, what type of ribosome is that? Everybody say bound ribosome. Bound ribosome, which will make proteins by assembling amino acids and forming peptide bonds. And because it's attached, that protein is going to, everybody say, leave the cell. Leave the cell. So it puts it inside of a vesicle. Everybody say vesicle. Vesicle. That's the first packaging. The Golgi apparatus repackages it again into another package, another set of vesicles. We'll just think of it as two packages. And then it can be sent outside of the cell in a process called exocytosis. So this endoplasmic reticulum is divided into two portions. I just talked about the rough endoplasmic reticulum, the one that contains the bound ribosomes. Some parts of the endoplasmic reticulum do not have any ribosomes attached. They are called smooth endoplasmic reticulum. You can't touch them, but they look smooth under a microscope. That's all it means. The rough ones look rough because they have ribosomes attached to them. So if I look over here, I have rough endoplasmic reticulum that has bound ribosomes, and then over here I have smooth endoplasmic reticulum, which does not have any ribosomes attached. So what's the difference? Well, the rough endoplasmic reticulum is obviously going to be packaging the proteins that are made by the ribosomes that are found on them, the bound ribosomes. The smooth has a different job. The smooth makes more of the phospholipid bilayer that your cells are made of, and it also breaks down toxins. Pretty important to be able to break down toxins? Probably. Okay. So smooth, even though it doesn't have ribosomes, still has a very important job. It's also where cholesterol and a few other things are made, but don't worry about that. Yeah. Did you know animals are the only thing that has cholesterol? Isn't that weird? Plants don't. So the portion of the ER with attached ribosomes is what is called rough endoplasmic reticulum because it has a rough appearance when viewed with an electron microscope. It's not that you can touch it. Remember, it's part of a cell. You can't touch anything that small. So the portion of ER with no attached to ribosomes is called smooth ER because it has a smooth appearance when viewed with an electron microscope. And the ribosome on the rough ER makes proteins that are packaged into vesicles. What are vesicles? Fat bubbles. They're made of phospholipids. They're like a little miniature cell membrane. And enzymes of the smooth ER make lipids and break down toxic substances. So the smooth ER has a very important job to make more of your phospholipids for your cell membrane and to break down toxins. And then, of course, that second structure we talked about, the Golgi apparatus. This is a set of flattened membrane-bound sacs. And the Golgi apparatus helps modify, sort, and package cell products for distribution. I think of this as the box that you put the bags of chips in after the endoplasmic reticulum packages them. So you can see this is the Golgi apparatus. So after the endoplasmic reticulum packages a protein, it then sends it to the Golgi apparatus. The Golgi apparatus puts it inside of a little fat sac here, and it pinches off. And then that little bubble, that vesicle, will float to the cell surface. And it will actually fuse with the cell, and it will release its contents outside of the cell. That's called exocytosis. It's kind of how cells, well, it's how cells go to the bathroom. It's also how they export proteins. So the ribosomes located on the rough ER make proteins which then cross into the membranes of the ER, and that's the endoplasmic reticulum. And the endoplasmic reticulum membrane then pinches off and forms a vesicle around the protein. That's the first package. Vesicles transport the proteins from the rough ER to the Golgi apparatus, where they are modified by enzymes and repackaged into new vesicles, like putting your bags inside of boxes. And these new vesicles transport the modified proteins to the cell membrane to be released outside of the cell through a process called exocytosis. I actually want you to write that down. Anytime your cell releases something, it's called exocytosis through a vesicle. So if I can get rid of things in vesicles, do you suppose I can bring them in vesicles as well? Yes. Let's talk about that. There's two times where this happens. Okay. I talked about exocytosis, things leaving, right? Let's talk about the other one. So exocytosis is exiting by a vesicle. That means there must be something else called endocytosis. And that would be entering, you should be writing this down, by a vesicle. And there's two types. There's penocytosis. And that's when a cell brings in small particles, and you can think of it as drinking. And then there is what is called phagocytosis. Which is large particles. And that's like a cell eating. So exocytosis, kind of like a cell going to the bathroom. It's like a cell getting rid of a protein. It can be a lot of different things. They can get rid of waste this way. They can expel used, worn-out parts, things they don't need, or they can release proteins in the body, in the bloodstream. And then there's endocytosis, where you bring things in. That's how cells eat or drink, bring in nutrients, because they must have a way to get food particles into the cell, right? They do this through endocytosis, and there's two types of endocytosis: penocytosis, which is where they bring in small particles, where the cell drinks, and then there's phagocytosis, which are large particles and to eat. Peno means small, phago means to eat in Latin. Cyto is cell, the process of a cell bringing in small particles. Penocytosis, cytoosis, the process of a cell, phago, eating. Yes? Golgi apparatus? Yes, ma'am. Yep. Did you guys get all that? So this, what is this process here? I'm just gonna don't say anything yet, but let's just look at this process. Let's just pretend that, you know, this is going out of the cell. See, it's being released out of the cell. What do we call? I gotta ask a question here. What do we call this process? What do we call this process? Come on. Let's go. It's okay to spell it wrong. Let's see what we're getting. Yay, exocytosis. Yay. I has no idea. Okay. Yes, it is exocytosis. How do I know? Because it's leaving, right? Anytime it's exiting the cell, this could be proteins, it could be waste. This kind of looks like waste. Kind of how cells go to the bathroom. Now, there are other structures in cells. Sometimes vesicles stay in cells. So vesicles help maintain homeostasis by storing and releasing a variety of substances as the cells need them. And a lysosome is a vesicle produced by the Golgi apparatus that contains enzymes that break down large molecules. And lysosomes can recycle old or damaged organelles and digest food particles to provide nutrients for the cell. So they're almost like the stomachs of the cell. They're floating around. There's little fat bubbles that hold enzymes in them. And then if there's an old part in my cell that's not working great, they'll actually sort of engulf it, bring it into them, and then they'll digest it. They'll break it down. And then they will float to the cell surface and release all of that by exocytosis, right? And so we have these lysosomes that are basically just more vesicles floating around. There are other things called food vacuoles when you bring things in through phagocytosis, and sometimes a food vacuole will fuse with a lysosome to digest the food that the cell brought in. Does that make sense? Let me draw it out. I know that's kind of a lot. So over here, I'm inside of the cell. So let's say this is food. So this is a food vacuole. Okay? And over here, let's say this is a lysosome. And inside the lysosome, there's this enzyme which breaks things down. I don't know. That's kind of a bad drawing. So what this lysosome would do is it would actually fuse. These two membranes would fuse, and they'd become part of each other. And this would actually engulf it. And this enzyme would break down the food. You get that? I know it's a terrible drawing. I'm not an artist. You guys know that, so shouldn't have me either. Okay. So a vacuole is a fluid-filled vesicle found in the cytoplasm of many plant cells. So as animals, we have vacuoles. They're called food vacuoles. Plants have a large vacuole. Do you remember last week what we called that large vacuole? What is the name of the large vacuole that plants have? It has a special name. Remember we talked about it being their water source, so that they don't have to run and get food because they can't run. Can't even walk. If they're a vine, they can crawl, though, right? So. Vines crawl. I guess it's not really crawling. See what you're coming up with. Central vacuole. Perfect, right off the bat. Yep. The large vacuole found in plant cells is called a central vacuole, and that is one of the major differences between plant and animal cells. So plants contain a large compartment called the central vacuole, which stores water, ions, nutrients, and waste. When water fills the central vacuole, the cell becomes rigid, allowing the plant to stand up. Now, when the vacuole loses water, the cell shrinks and the plant wilts. So we've seen this, right? Hot, dry summer day, a flower. It wilts, right? You can see it droop, okay? Well, that water loss, it's called turgor pressure. When you lose that turgor pressure, the plant becomes limp and bends over. If you water the plant, it becomes firm and stands up again because you increase the turgor pressure. And so this is why plants like an environment that contains lots of water. Now, some protists have what are called contractile vacuoles. You guys remember looking at the pond water and we saw things scooting around? Imagine this. Imagine being so simple, you're like a single cell, and you basically sucked water in your mouth and shot it out your butt, and that's how you moved around. Like your body was this big muscle that just squirted yourself. That's what they do. They bring water in their mouth and they squirt it out their back. They're like a little jet, and that's what moves them through the water. They're called rotifers, okay? So a food vacuole is another type of vacuole. It's formed when the cell membrane surrounds food particles outside of the cell, and they bring them in through what process would I bring food into my cell? What is that process called? What process, be specific, would I use to bring food into my cell? What process would I use to bring food in and create a food vacuole? It sounds like a test question. What process would create a food vacuole? Yay! It's a type of endocytosis called phagocytosis. Why is it phago and not pino? Because phago means to eat, right? Pino is to drink small particles. Endoritis. What is it? Endorotis? Fascitosis. Fascitosis? I don't know. Anyway, you got it. Close. That's the pop. Okay. Nope. So cells need a constant source of energy. The energy for cellular functions is produced by chemical reactions that occur in what is called the mitochondria and chloroplasts. Chloroplasts are only found in plant cells. Mitochondria are found in both plant and animal cells. In both organelles, chemical reactions produce adenosine triphosphate, the form of energy that fuels almost all cell processes. So chloroplasts, they make a little bit of ATP, but they also make glucose. Mitochondria take the glucose, and they make a lot of ATP. So let me tell you something kind of cool about mitochondria and chloroplasts. You guys, you know how you're half your mom, half your dad? Kind of. What if I told you you're more your mom than your dad? So, well, here's why. And it has nothing to do with what you look like. So inside of every one of your cells, we all have mitochondria. You guys? And your mitochondrial DNA is not half mom, half dad. It is 100% moms. Okay? Remember how I showed you the coral bleaching video? Okay, so imagine we have a symbiote inside of all of our cells, and it sort of came to us by an infection from our mother's egg. Okay? It's called a mitochondria. And the mitochondria is what makes all of your ATP, and it comes from your mom. It does not come from your dad. Here's the other creepy thing. The mitochondria divides on its own. It goes through what's called binary fission. That's how bacteria reproduce. It has its own DNA. It divides independently of your cell, and it has its own ribosomes, and it makes its own proteins. And it's 100% from your mom. Basically clones itself. What I mean by this is you have the exact same mitochondrial DNA as your brother or sister. You have the exact same mitochondrial DNA as your mom and as your mom's brothers and sisters because they got it from their mom. So you'll be identical. When there are things called, you ever heard of mass genocide? They come in and try to kill off. So a lot of times they find babies. Can a baby say that's my aunt over there in Zimbabwe? I say that because that's where most of the mass genocides happen in Africa. But can they say that? They're a baby. No. So they can take their mitochondrial DNA and match them up with a relative so that they aren't like an orphan. I mean, they are an orphan, but at least with family that way. They did that in Hurricane Katrina, too. And so I think it's really creepy just the fact that we have this little independent thing inside of us that we are not more than one cell without. And it comes from your mom. Now, we'll get into this later. It's called the endosymbiotic theory. But it's also thought that this used to be a bacteria because the DNA of the mitochondria is bacterial DNA. And so is chloroplasts. So, yeah. Of what? You need the other half of the rest of us. Yeah. Well, it's a good question. So what's the point of having a dad? Well, we are, at this point, so far evolved, we aren't going to rewind the clock to not have men. But what do men do for a species? They create diversity, which helps drive natural selection. It makes us more diverse. It makes us more apt to survive harsh conditions. Okay? Now, if I look at things like amphibians, like frogs, they don't need a male. If a male's there, it's great, but if they're not, they will self-fertilize, and the frog can go ahead and have healthy frogs without the male. So alligators can do that too. I don't think you usually don't rewind evolution. It usually only goes for one way. You can't rewind evolution, I guess is what I'm trying to tell you. It only goes in one direction. Kind of like the Bad Boy Band, right? No. So a chloroplast is an organelle found in plant and algae cells that uses light energy to make carbohydrates from carbon dioxide and water. So chloroplasts, just like mitochondria, they reproduce like bacteria. They have bacterial DNA. And the chloroplast is the same as the mother. And you go, wait a minute, Mr. Mason. Plants are not boys and girls, and I say baloney. They are too. Okay? So we just dissected flowers actually in AP Bio. I wish I'd have kept some. But plants have ovaries, right? Flowers, at the base of every flower, there is what is called an ovary, and inside of that are eggs. And then the pollen is the sperm, right? And so there is sex in flowers, okay? So it sounds weird, but it's called the birds and the bees for a reason, right? Yeah. So even a chloroplast is only from the female egg portion of the flower, okay? Which is really, now you guys are really starting to think about flowers differently. So chloroplasts are surrounded by two membranes and have several stacks of flattened sacs. They're called thylakoids. We'll spend a whole chapter talking about those, where energy production takes place. Now, plant cells may have several chloroplasts, and usually they do. The greener they are, the more chloroplasts they have. What's a structure on plants that would have a lot of chloroplasts in their cells? What's a structure on plants that would have a lot of chloroplasts in the cells? Leave me alone, Mr. Mason. Not the roots. No, the roots are not green. And we talked briefly about the mitochondria, but these are cell organelles that use energy from organic compounds to make ATP. Most of the ATP needed by a cell is produced inside of mitochondria, and both animal and plant cells contain mitochondria. You guys, we are not able to be more than one cell without mitochondria. We just don't have enough energy. So a smooth outer membrane and a folded inner membrane surround a mitochondrion, and ATP is produced by enzymes on the folds of the inner membrane. So this is a mitochondrion. I want to talk about these folds because they are very important. Without this fold, something very weird would happen. So this is where we add hydrogen and oxygen. That will yield energy and water. You guys remember the Hindenburg? I don't. I wasn't born yet. You guys remember hearing about the Hindenburg? Okay. You're like a 10-year senior. Or 20. So the Hindenburg was this balloon. They said, let's fill this thing with like a very volatile chemical or gas called hydrogen. Sounds great. It's lighter than air. It floats. The only problem about hydrogen, it's like the ADD child of the periodic table. It likes to bond with everything. Bounces around. Yeah, like bonding means boom, right? Okay? We now use what? Helium, because it doesn't bond with anything, right? So, but they did use hydrogen. So if I were going to bring hydrogen and oxygen together inside of a structure inside of my body, should I isolate that reaction? Yeah, and that's what these folds do. See how there's a bunch of these folds? They basically isolate the chemical reactions. Because if these were all next to each other, I mean, we wouldn't blow up, but we wouldn't be here. We wouldn't be more than one cell. We would have never gotten past that point. So you need these folds. These folds are what are called cristae. Okay? And those cristae, those folds in the mitochondria, are what allow them to produce all of this ATP. Well, I'll show you what this reaction does. We'll make a little hydrogen bomb in here. Please don't pack up right now. It's not that time. By the way, do all cells have a cell membrane? Is a cell membrane 2 layers? Is this 2 layers? Yes. Yes. Okay? So, listen, any organelle inside of a cell that has two layers has its own DNA, divides on its own, has its own ribosomes, and is bacterial DNA. That's kind of creepy, isn't it? It is like a cell in a cell. In fact, we'll learn later what's called the endosymbiotic theory, which is how they came to be, where one bacteria engulfed another, and instead of destroying it, they formed a relationship, and this is how you end up with multicellular organisms. So the cytoskeleton helps the cell move, keeps its shape, and organizes its parts. DNA instructions are copied as RNA messages, which leave the nucleus. In the cytoplasm, ribosomes use the RNA messages to assemble proteins. And the endoplasmic reticulum and Golgi apparatus are organelles involved in preparing proteins for extracellular export. Do not forget about exocytosis and endocytosis, pinocytosis, phagocytosis. The first packaging is done by the endoplasmic reticulum, the second one by the Golgi apparatus. Vesicles help maintain homeostasis by storing and releasing a variety of substances as the cells need them. Don't forget about lysosomes, helping break things down, and the food vacuoles, the contractile vacuoles that help some organisms move. And the energy for cellular function is produced by chemical reactions that occur in the mitochondria and chloroplast, each of which has its own DNA, each has its own ribosomes, each make their own proteins. Both give us energy. We'll spend a chapter on both of them.